Chapter 4: Protein Structure and Function. When we look at a cell through a microscope or analyze its electrical or biochemical activity, we are, in essence, observing the handiwork of proteins. Proteins are the building blocks from which cells are assembled, and they constitute most of the cell's dry mass. But in addition to providing the cell with shape and structure, proteins also execute nearly all its myriad functions. Enzymes promote intracellular chemical reactions by providing intricate molecular surfaces, contoured with particular bumps and crevices that can cradle or exclude specific molecules. Proteins embedded in the plasma membrane form the channels and pumps that control the passage of nutrients and other small molecules into and out of the cell. Other proteins carry messages from one cell to another, or act as signal integrators that relay information from the plasma membrane to the nucleus of individual cells. Yet others serve as tiny molecular machines with moving parts. Some proteins, such as kinesin, propel organelles through the cytoplasm. Others, such as helicases, pry open double-stranded DNA molecules. Specialized proteins also act as antibodies, toxins, hormones, antifreeze molecules, elastic fibers, or luminescence generators. Before we can hope to understand how genes work, how muscles contract, how nerves conduct electricity, how embryos develop, or how our bodies function, we must understand proteins. The multiplicity of functions carried out by proteins, panel 4.1, page 120, arises from the huge number of different shapes they adopt. So we begin our description of these remarkable macromolecules by discussing their three-dimensional structures and the properties that these structures confer. We next look at how they work, how enzymes catalyze chemical reactions, how some proteins act as molecular switches, and how others generate coherent movement. We then examine how cells control the activity and location of the proteins they contain. Finally, we present a brief description of the techniques that biologists use to work with proteins, including methods for purifying them from tissues or cultured cells and determining their structures. The shape and structure of proteins. From a chemical point of view, proteins are by far the most structurally complex and functionally sophisticated molecules known. This is perhaps not surprising, considering that the structure and activity of each protein has been developed and fine-tuned over billions of years of evolutionary history. We start by considering how the position of each amino acid in the long string of amino acids that forms a protein determines its three-dimensional shape, a structure stabilized by non-covalent interactions between different parts of the molecule. Understanding protein structure at the atomic level will allow us to describe how the precise shape of each protein determines its function in the cell. The shape of a protein is specified by its amino acid sequence. Proteins, as you may recall from Chapter 2, are assembled from a set of 20 different amino acids, each with different chemical properties. A protein molecule is made from a long chain of these amino acids, each linked to its neighbor through a covalent peptide bond. Figure 4.1. Proteins are therefore referred to as polypeptides or polypeptide chains. In each type of protein, the amino acids are present in a unique order, called the amino acid sequence, which is exactly the same from one molecule of that protein to the next. One molecule of insulin, for example, has the same amino acid sequence as every other molecule of insulin. Many thousands of different proteins have been identified. Each has its own distinct amino acid sequence. Each polypeptide chain consists of a backbone that supports the different amino acid side chains. The polypeptide backbone is made from the repeating sequence of the core atoms of the amino acids that form the chain. Projecting from this repetitive backbone are any of the 20 different amino acid side chains, the parts of the amino acids that are not involved in forming the peptide bond. Figure 4.2. These side chains give each amino acid its unique properties. Some are nonpolar and hydrophobic, water-fearing. Some are negatively or positively charged. Some are chemically reactive, and so on. The atomic structures of the 20 amino acids in proteins are presented in Panel 2.5, pages 72 to 73, and a brief list of amino acids with their abbreviations is provided in Figure 4.3. Long polypeptide chains are very flexible. Many of the covalent bonds that link carbon atoms in an extended chain of amino acids allow free rotation of the atoms they join. Thus, proteins can in principle fold in an enormous number of ways. Each folded chain is constrained by many different sets of weak non-covalent bonds that form within proteins. These bonds involve atoms in the polypeptide backbone as well as atoms in the amino acid side chains. The non-covalent bonds that help proteins maintain their shape include hydrogen bonds, electrostatic attractions, and van der Waals attractions, which are described in Chapter 2. See Panel 2.7, pages 76 to 77. Because individual non-covalent bonds are much weaker than covalent bonds, it takes many non-covalent bonds to hold two regions of a polypeptide chain together tightly. The stability of each folded shape will therefore be affected by the combined strength of large numbers of non-covalent bonds. Figure 4.4. A fourth weak force, hydrophobic interaction, also plays a central role in determining the shape of a protein. In an aqueous environment, hydrophobic molecules, including the nonpolar side chains of particular amino acids, tend to be forced together to minimize their disruptive effect on the hydrogen-bonded network of the surrounding water molecules. See Panel 2.2, pages 66 to 67. Therefore, an important factor governing the folding of any protein is the distribution of its polar and nonpolar amino acids. The nonpolar hydrophobic side chains, which belong to amino acids such as phenylalanine, leucine, valine, and tryptophan, tend to cluster in the interior of the folded protein, just as hydrophobic oil droplets coalesce to form one large droplet. Tucked away inside the folded protein, hydrophobic side chains can avoid contact with the aqueous cytosol that surrounds them inside a cell. In contrast, polar side chains, such as those belonging to arginine, glutamine, and histidine, tend to arrange themselves near the outside of the folded protein, where they can form hydrogen bonds with water and with other polar molecules. Figure 4.5. When polar amino acids are buried within the protein, they are usually hydrogen bonded to other polar amino acids or to the polypeptide backbone. Figure 4.6. Proteins fold into a conformation of lowest energy. Each type of protein has a particular three-dimensional structure, which is determined by the order of the amino acids in its chain. The final folded structure or conformation adopted by any polypeptide chain is determined by energetic considerations. A protein generally folds into the shape in which the free energy, G, is minimized. See page 91. Protein folding has been studied in a laboratory using highly purified proteins. A protein can be unfolded or denatured by treatment with solvents that disrupt the non-covalent interactions holding the folded chain together. This treatment converts the proteins into a flexible polypeptide chain that has lost its natural shape. When the denaturing solvent is removed, The protein often refolds spontaneously or renatures into its original conformation. Figure 4.7. The fact that a renatured protein can, on its own, regain the correct conformation indicates that all the information necessary to specify the three-dimensional shape of a protein is contained in its amino acid sequence. Each protein normally folds into a single stable conformation. This conformation, however, often changes slightly when the protein interacts with other molecules in the cell. This change in shape is crucial to the function of the protein, as we shall see later in this chapter. When proteins fold incorrectly, they sometimes form aggregates that can damage cells and even whole tissues. Aggregated proteins underlie a number of neurodegenerative disorders, including Alzheimer's disease and Huntington's disease. Prion diseases, such as scrapie in sheep, bovine spongiform encephalopathy, BSE or mad cow disease in cattle, and Creutzfeldt-Jakob disease, CJD in humans, are also caused by protein aggregates. The prion protein, PRP, can adopt a misfolded form that is considered infectious because it can convert properly folded PRP proteins in the infected brain into the abnormal conformation. Figure 4.8. This allows the misfolded form of PRP to spread rapidly from cell to cell, causing the death of the infected animal or human. Although a protein chain can fold into its correct conformation without outside help, protein folding in a living cell is generally assisted by special proteins called molecular chaperones. These proteins bind to partly folded chains and help them to fold along the most energetically favorable pathway, as we shall discuss in Chapter 7. Chaperones are vital in the crowded conditions of the cytoplasm because they prevent newly synthesized protein chains from associating with the wrong partners. Nevertheless, the final three-dimensional shape of the protein is still specified by its amino acid sequence. Chaperones merely make the folding process more efficient and reliable. Proteins come in a wide variety of complicated shapes. Proteins are the most structurally diverse macromolecules in the cell. Although they range in size from about 30 amino acids to more than 10,000, the vast majority of proteins are between 50 and 2,000 amino acids long. Proteins can be globular or fibrous. They can form filaments, sheets, rings, or spheres. Figure 4.9 presents a sampling of proteins whose exact structures are known. We will encounter many of these proteins later in this chapter and throughout the book. Resolving a protein structure often begins with determining its amino acid sequence, a task that can be accomplished in several ways. For many years, protein sequencing was accomplished by directly analyzing the amino acids in the purified protein. The first protein sequenced was the hormone insulin in 1955. Today, we can determine the order of amino acids in a protein much more easily by sequencing the gene that encodes it, discussed in Chapter 10. Once the order of the nucleotides in the DNA that encodes a protein is known, this information can be converted into an amino acid sequence by applying the genetic code, discussed in Chapter 7. The amino acid sequences of millions of proteins have already been determined in this way, and they have been collected into vast electronic databases that allow users to obtain the amino acid sequence of any protein almost instantaneously. Although all the information required for a polypeptide chain to fold is contained in its amino acid sequence, we have not yet learned how to reliably predict a protein's detailed three-dimensional conformation, the spatial arrangement of its atoms from its sequence alone. At present, the only way to discover the precise folding pattern of any protein is by experiment, using either X-ray crystallography or nuclear magnetic resonance, NMR spectroscopy, as we discuss later in the chapter. So far, the structures of about 20,000 different proteins have been completely analyzed by these techniques. Most have a three-dimensional conformation so intricate and irregular that their structure would require an entire chapter to describe in detail. Because the structure of a large protein can be overwhelming to look at, we will illustrate the intricacies of protein conformation by examining the structure of a smaller protein domain. As we discuss shortly, most proteins are formed from multiple domains, each folding into a compact three-dimensional structure. In Panel 4.2, pages 128 to 129, we present four different depictions of SH2, a protein domain that, as part of proteins involved in cell signaling, has important functions in eukaryotic cells. Built from a string of 100 amino acids, the structure is displayed as A polypeptide backbone model, B, a ribbon model, C, a wire model that includes the amino acid side chains, and D, a space-filling model. As indicated in the panel, each model emphasizes different features of the polypeptide. The three horizontal rows show the SH2 domain in different orientations, and the images are colored to distinguish the path of the polypeptide chain, from its N-terminus, purple, to its C-terminus, red. We will describe the different structural elements in this protein domain shortly. From Panel 4.2, we can clearly see how amazingly complex protein conformation is, even for a small domain like SH2. To visualize such complicated structures, scientists have developed various graphical and computer-based tools that generate a variety of images of a protein, some of which are depicted in Panel 4.2. These images can be displayed on the screen and rotated to view all aspects of the structure. Movie 4.1. In addition, describing and presenting such complex protein structures is made easier by recognizing that several common folding patterns underlie these conformations, as we discuss next. The alpha helix and the beta sheet are common folding patterns. When the three-dimensional structures of many different protein molecules are compared, it becomes clear that although the overall conformation of each protein is unique, two regular folding patterns are often present. Both were discovered more than 50 years ago from studies of hair and silk. The first folding pattern to be discovered, called the alpha helix, was found in the protein alpha-keratin, which is abundant in skin and its derivatives, such as hair, nails, and horns. Within a year of the discovery of the alpha helix, a second folded structure, called a beta sheet, was found in the protein fibrin, the major constituent of silk. Biologists often use Greek letters to name their discoveries, with the first example receiving the designation alpha, the second beta, and so on. These two folding patterns are particularly common because they result from hydrogen bonds that form between the NH and C double bond O groups in the polypeptide backbone. Because the amino acid side chains are not involved in forming these hydrogen bonds, alpha helices and beta sheets can be generated by many different amino acid sequences. In each case, the protein chain adopts a regular, repeating form or motif. These structural features and the shorthand cartoon symbols that are often used to represent them in models of protein structures are presented in Figure 4.10. Helices form readily in biological structures. The abundance of helices in proteins in a way, not surprising. A helix is a regular structure that resembles a spiral staircase, as illustrated in Figure 4.11. It is generated simply by placing many similar subunits next to each other, each in the same strictly repeated relationship to the one before. Because it is very rare for subunits to join up in a straight line, this arrangement will generally result in a helix. Depending on the twist of the staircase, a helix is said to be either right-handed or left-handed. Figure 411e. Handedness is not affected by turning the helix upside down, but it is reversed if the helix is reflected in the mirror. An alpha helix is generated when a single polypeptide chain turns around itself to form a structurally rigid cylinder. A hydrogen bond is made between every fourth amino acid, linking the C double bond O of one peptide bond to the NH of another. See figure 410a. This gives rise to a regular helix with a complete turn every 3.6 amino acids. Movie 4.2. Short regions of alpha helix are especially abundant in the proteins located in cell membranes, such as transport proteins and receptors. We will see in Chapter 11 that those portions of a transmembrane protein that cross the lipid bilayer usually form an alpha helix that is composed largely of amino acids with nonpolar side chains. The polypeptide backbone, which is hydrophilic, is hydrogen bonded to itself in the alpha helix, and it is shielded from the hydrophobic lipid environment of the membrane by its protruding nonpolar side chains. Figure 412. Sometimes 2 or 3 alpha helices will wrap around one another to form a particularly stable structure known as a coiled coil. This structure forms when the alpha helices have most of their nonpolar hydrophobic side chains on one side, so that they can twist around each other with these side chains facing inward, minimizing their contact with the aqueous cytosol. Figure 413. Long, rod-like coiled coils form the structural framework for many elongated proteins. Examples include alpha-keratin, which forms the intracellular fibers that reinforce the outer layer of the skin, and myosin, the protein responsible for muscle contraction, discussed in Chapter 17. Beta sheets form rigid structures at the core of many proteins. SH2, the small protein structure we examined in Panel 4.2, contains both alpha helices and a beta sheet. Beta sheets are made when hydrogen bonds form between segments of polypeptide chains lying side by side. See figure 410d. When the structure consists of neighboring polypeptide chains that run in the same orientation, say from the N-terminus to the C-terminus, it is considered a parallel beta sheet. When it forms from a polypeptide chain that folds back and forth upon itself, with each section of the chain running in the direction opposite to that of its immediate neighbors, the structure is an anti-parallel beta sheet. Figure 414. Both types of beta sheet produce a very rigid, pleated structure, and they form the core of many proteins. SH2, for example, has an anti-parallel beta sheet core. Beta sheets have remarkable properties. They give silk fibers their extraordinary tensile strength, and they can help to keep insects from freezing in the cold. In an antifreeze protein isolated from a beetle that lives in cold climates, a series of parallel beta sheets forms a beautifully flat surface along one side of the protein molecule. Figure 415. This array appears to offer a perfect platform for binding to the regularly spaced water molecules that are present in an ice lattice. By adhering to the ice crystals that form when water is cooled below its freezing point, the antifreeze protein prevents the ice crystals from growing, thereby keeping the insect cells from freezing solid. Proteins have several levels of organization. A protein structure does not end with alpha helices and beta sheets. There are additional levels of organization. These levels are not independent, but are built one upon the next until the three-dimensional structure of the entire protein has been fully defined. A protein structure begins with its amino acid sequence, which is thus considered its primary structure. The next level of organization includes the alpha helices and beta sheets that form within certain segments of a polypeptide chain. These folds are elements of the protein's secondary structure. The full three-dimensional conformation formed by an entire polypeptide chain, including the alpha helices, beta sheets, random coils, and any other loops and folds that form between the N and C termini, is sometimes referred to as the tertiary structure. See the structure shown in panel 4.2 for example. Finally, if a particular protein molecule is formed as a complex of more than one polypeptide chain, then the complete structure is designated its quaternary structure. Studies of the conformation, function, and evolution of proteins have also revealed the importance of a level of organization distinct from those just described. This organizational unit is the protein domain, which is defined as any segment of a polypeptide chain that can fold independently into a compact, stable structure as we have seen in the case of the SH2 domain. See panel 4.2, pages 128 to 129. A domain usually consists of 100 to 250 amino acids, folded into alpha helices and beta sheets, and other elements of secondary structure. And it is a modular unit from which many larger proteins are constructed. Figure 4.16. The different domains of a protein are often associated with different functions. For example, the bacterial catabolite activator protein, CAP, illustrated in Figure 4.16, has two domains. The small domain binds to DNA, while the large domain binds cyclic AMP, an intracellular signaling molecule. When the large domain binds cyclic AMP, it causes a conformational change in the protein that enables the small domain to bind to a specific DNA sequence and promote the expression of adjacent genes. The SH2 domain, see panel 4.2, is found in many different polypeptides, where it serves as a binding domain to promote protein-protein interactions, as we shall see in Chapter 16. Small protein molecules, such as the oxygen-carrying muscle protein myoglobin, contain only a single domain. See Figure 4.9. Larger proteins can contain as many as several dozen domains, which are usually connected by relatively unstructured lengths of polypeptide chain. Ribbon models of three differently organized domains are presented in Figure 4.17. Few of the many possible polypeptide chains will be useful. In theory, a vast number of different polypeptide chains can be made. Because each of the 20 amino acids is chemically distinct and each can, in principle, occur at any position in a protein chain, a polypeptide chain 4 amino acids long has 20 times 20 times 20 times 20 equals 160,000 different possible sequences. In other words, for a polypeptide that is n amino acids long, 20 to the power of n different chains are possible. For a typical protein length of 300 amino acids, more than 20 to the power of 300, that's 10 to the power of 390, structurally different polypeptide chains could theoretically be made. However, only a very small fraction of this unimaginably large number of polypeptide chains would fold into a stable, well-defined, three-dimensional conformation. The vast majority of individual protein molecules would have many different conformations of roughly equal stability, each conformation having different chemical properties. So why do most proteins present in cells adopt unique and stable conformations? The answer is that a protein with many different conformations and variable properties would not be biologically useful, for it would be like a tool that unexpectedly changes its function. Such proteins would therefore have been eliminated by natural selection through the long trial and error process that underlies evolution, discussed in Chapter 9. Because of natural selection, the amino acid sequences of most present-day proteins have evolved to guarantee that the polypeptide will adopt an extremely stable conformation, a structure that bestows upon the protein the exact chemical properties that will enable it to perform a particular catalytic or structural function in the cell. Proteins are so precisely built that the change of even a few atoms in one amino acid can sometimes disrupt the structure of a protein and thereby eliminate its function. In fact, many protein structures are so stable and effective that they have been conserved throughout evolution among many diverse organisms. The three-dimensional structures of the DNA binding domains from the yeast alpha-2 protein and the Drosophila engrailed protein, for example, are almost completely superimposable, even though these organisms are separated by more than 1 billion years of evolution. Proteins can be classified into families. Once a protein had evolved a stable conformation with useful properties, its structure could be modified over time to enable it to perform new functions. We know that this occurred quite often during evolution, because many present-day proteins can be grouped into protein families, in which each family member has an amino acid sequence and a three-dimensional conformation that closely resembles that of the other family members. Consider, for example, the serine proteases, a family of protein-cleaving proteolytic enzymes that includes the digestive enzymes chymotrypsin, trypsin, and elastase, as well as several proteases involved in blood clotting. When any two of these enzymes are compared, portions of their amino acid sequences are found to be nearly the same. The similarity of their three-dimensional conformations is even more striking. Most of the detailed twists and turns in the polypeptide chains, which are several hundred amino acids long, are virtually identical. Figure 418. The various serine proteases nevertheless have distinct enzymatic activities, each cleaving different proteins or the peptide bonds between different types of amino acids. Slight differences in structure allow each of these proteases to prefer different substrates. Thus, each carries out a distinct function in an organism. Large protein molecules often contain more than one polypeptide chain. The same weak non-covalent bonds that enable a polypeptide chain to fold into a specific conformation also allow proteins to bind to each other to produce large structures in the cell. Any region on a protein surface that interacts with another molecule through sets of non-covalent bonds is termed a binding site. A protein can contain binding sites for a variety of molecules, large and small. If a binding site recognizes the surface of a second protein, the tight binding of two folded polypeptide chains at this site will create a larger protein whose quaternary structure has a precisely defined geometry. Each polypeptide chain in such a protein is called a subunit. Each of these protein subunits may contain more than one domain, a portion of the polypeptide chain that folds up separately. In the simplest case, two identical folded polypeptide chains form a symmetrical complex of two protein subunits, called a dimer, that is held together by interactions between two identical binding sites. The cap protein in bacterial cells is a dimer, figure 419a, that is formed from two identical copies of the protein subunit shown previously in figure 416. Many other symmetrical protein complexes formed from multiple copies of a single polypeptide chain are commonly found in cells. The enzyme neuraminidase, for example, consists of a ring of 4 identical protein subunits, figure 419b. Other proteins contain two or more different types of polypeptide chains. Hemoglobin, the protein that carries oxygen in red blood cells, is a particularly well-studied example. Figure 420. The protein contains two identical alpha-globin subunits and two identical beta-globin subunits, symmetrically arranged. Many proteins contain multiple subunits, and they can be very large. Movie 4.5. Proteins can assemble into filaments, sheets, or spheres. Proteins can form even larger assemblies than those discussed so far. Most simply, a chain of identical protein molecules can be formed if the binding site of one protein molecule is complementary to another region on the surface of another protein molecule of the same type. Because each protein molecule is bound to its neighbor in an identical way, the molecules will often be arranged in a helix that can be extended indefinitely. Figure 421. This type of arrangement can produce an extended protein filament. An actin filament, for example, is a long helical structure formed from many molecules of the protein actin. Figure 422. Actin is extremely abundant in eukaryotic cells, where it forms one of the major filament systems of the cytoskeleton, discussed in Chapter 17. Other sets of proteins associate to form extended sheets or tubes, as in the microtubules of the cell cytoskeleton. Figure 423. Or cage-like spherical shells, as in the protein coats of virus particles. Figure 424. Many large structures, such as viruses and ribosomes, are built from a mixture of one or more types of proteins plus RNA or DNA molecules. These structures can be isolated in pure form and associated into their constituent macromolecules. It is often possible to mix the isolated components back together and watch them reassemble spontaneously into the original structure. This demonstrates that all the information needed for assembly of the complicated structure is contained in the macromolecules themselves. Experiments of this type show that much of the structure of a cell is self-organizing. If the required proteins are produced in the right amounts, the appropriate structures will form. Some types of proteins have elongated fibrous shapes. Most of the proteins we have discussed so far are globular proteins, in which the polypeptide chain folds up into a compact shape like a ball with an irregular surface. Enzymes tend to be globular proteins, even though many are large and complicated with multiple subunits. Most have a quaternary structure with an overall rounded shape. See Figure 4.9. In contrast, other proteins have roles in the cell that require them to span a large distance. These proteins generally have a relatively simple, elongated, three-dimensional structure and are commonly referred to as fibrous proteins. One large class of intracellular fibrous proteins resembles alpha-keratin, which we met earlier. Keratin filaments are extremely stable, long-lived structures such as hair, horn, and nails are composed mainly of this protein. An alpha-keratin molecule is a dimer of two identical subunits, with the long alpha helices of each subunit forming a coiled coil. See Figure 4.13. These coiled coil regions are capped at either